Martha Carlin is big into the world of the microbiome as it relates directly to her husband's Parkinson's diagnosis. And so as she has took on, the experience of dealing with adversity, she's come up with solutions, and she is a wealth of information, wonderful person. So we hit on that at the first, and then the second part, we dig into kind of an introductory level with some topics that can get really confusing, but I think they're of the utmost importance. Martha, thank you so much for jumping on here. I've been excited to visit with you for a while. You're somebody that I can relate to in that a loved one was faced with something extreme. In your case, your husband with Parkinson's. In my case, my son with stage 4 cancer. And how do we deal with this? And so in trying to piece all of this together, the different bodies of work, the contradictions across everything start to play, the opinions. And so in a way, I feel like that's a positive because it made me try to find the fundamentals of what is actually going on instead of just parroting a narrative. And so if you would, just give that brief little intro to what you're doing. And I just want to jump into the nitty gritty.
Sure. So 22 years ago, actually, 2002, my husband was diagnosed with Parkinson's, and he was 44 years old. We had 2 small children, and then I had a little bit older child from another marriage. And, he had been a healthy, marathon running, active, never did anything bad, kind of. It just didn't make sense to me. And my background was actually I had a degree in accounting, which is a system that has to balance. And I came out of college and was trained as an auditor. And they train you as an auditor, never accept anything at face value, examine the evidence for yourself. And then I was taught a way to evaluate business risk called transaction flow review, where you map out all of the processes of a business and who touches them and what each step of the process is. So you draw these big flow charts and you interview people looking for breakpoints in the system because that's where the business risk is. So when John was diagnosed with Parkinson's, I said to myself, well, they give you a drug and just sort of send you on your way. And I said, I think this is a systems problem. So I need to apply my transaction flow review process to understanding what's going on. And so what's the main thing flowing through the body? Well, so the first place I looked was food and water. And I threw out everything in the pantry and started over with, fresh food and cooking as much as possible. I mean, I still traveled a lot. So, but that kind of started me on the path of digging into the science of food, how we grow our food, the chemicals that are used on food, and how that was impacting us. And then, fast forward from 2002 to 2014, so 12 years later, I had been studying lots about the food supply and the, basic kind of physiology of what's going on in Parkinson's. And then I got a book called Missing Microbes by Dr. Martin Blaser, and heard this term, the microbiome, which I had never heard of before. And he's talking about the age of antibiotics and, how it's been overprescribed since we've discovered them, and then how much they're used in the food supply and what that's done in the rise of chronic disease. And I said, oh, that makes so much sense because I knew my husband had had a history of chronic strep infections as a kid. And so he'd taken a lot of antibiotics. And about 6 months later, this researcher, Philip Schepperhans, published the first research paper that showed he could divide there's 2 primary types of Parkinson's. One is people have tremor dominant, and the other is they have the curved posture and freezing gait problems. And he could actually divide that by the bacteria in the gut. And I was like, that's it. the gut is the general ledger of the body, and it's got to balance. And so I quit my job and started funding some research at the University of Chicago. And from that, actually went on to found the Bio Collective to collect people's poop to look at the microbiome. And I founded that company with Dr. Jack Gilbert from the University of Chicago and Dr. Suzanne Vernon, who was a virologist who had worked at the CDC for about 17 years.
So with that, Martha, what have you found in the microbiome that is really kind of wowed you?
Oh gosh, we found so many interesting things in the microbiome, but in our work with Parkinson's, we found some connections to. I actually did some digging around and found some connections potentially to glyphosate exposure, or that the actual target gene, which is an AROA gene, is more prevalent in people with Parkinson's than it is with people without Parkinson's. So the target gene that the herbicide works against, it was more prevalent in people with Parkinson's. So that was kind of interesting. But we did, over the course of 5 years, we did sampling of not just people with Parkinson's, but people across the population. But in our Parkinson's research, then we took the data and we did some machine learning with a company that typically looks for cancer biomarkers, but they used our genomic data, and they found some biomarkers related to mycobacteria, which is also interesting because there is some evidence that in the Harvard, there's a Harvard doctors and nurses study that has a connection to drinking low-fat milk or eating low-fat dairy with increased risk of Parkinson's. And the milk supply, I think it's fairly well known that the cows are infected with something called Mycobacterium avium paratuberculosis, and that has some connection to IBD. And about a third of people with IBD go on to develop Parkinson's. So there's some interesting kind of pieces of the puzzle there. And mycobacteria has a tendency over time, over the life of the infection, to alter metabolism of carbohydrates and later on metabolism of lipids as the expansion of the infection goes on, I would say. And then we also found biomarkers related to Haemophilus influenza. So Haemophilus influenza, there's a number of genes involved with that are transposable elements. I'll say, I don't want to get like too technical on the genetics of things, but that could actually, and I don't want to get in trouble for saying this, but could actually potentially go back to, that's a vaccine that we get, Haemophilus influenza B, I think Hib B. So there could be some connection to that, and maybe potentially having some effect that's causing an autoimmune reaction or something. So, and there is some evidence in, I think there's a Polish study that has a connection to some of that. And I have if you go to my YouTube channel, the BiotiQuest Probiotics YouTube channel, I have a presentation that I did on molecular mimicry, and that has these connections between little proteins that bacteria make that are similar to proteins in human tissues. And so if your immune system reacts to the protein from the bacteria, it can make an antigen that then is looking around the body for something similar, and you have a tissue that's similar. And in Parkinson's, there are, there's research showing elevated antibodies to all kinds of strange things. Anti-brain lysate, anti-double-stranded DNA, anti-cardiolipin, which is the lipid membrane of the mitochondria, anti-phospholipid, anti-phosphatidylserine. So there's a long list of different research projects that have shown different elevated antibodies, and you can map some of those, which I've done in that presentation, to different bacterial proteins.
Wow, I'll have to go do that. So, in essence, it is. Would that be a metabolite that the bacteria produces that is the body basically doesn't like and attacks it, as well as the biomimic proteins within the human body?
I mean, basically, yeah, that's
Okay. One of the very first things that was eye opening for me on the microbiome was some of the immunotherapy studies were dependent on if a certain strand of bacteria was present or not, if the immunotherapy was effective or not. And so that got me really trying to think. And so what I did was just tried to find what was the best probiotic I could get. And so as you have gone through that, how are you able to categorize the bacteria we're going to get? Because it was my understanding it's very difficult to get colonization of the keystone species, so to speak, with food. So what are your way of laying out getting the gut back optimized?
well. So we started. Initially, I didn't think I was going to be making probiotics, but in about 2016. So that's 6 years. Is that 8 years ago now? Is that right? 2016. I went to a Parkinson's conference, and there were some researchers there presenting data showing that the sugar alcohol mannitol could stop the aggregation of the proteins that were implicated in Parkinson's. So I came back and I got a mannitol chemistry book, started reading that and all the different things that this sugar alcohol did. But there was also a chapter in there about bacteria that use it as a carbon source or produce it. And so that gave me the idea that kind of went on to inform the whole lineup that we have at BiotiQuest, but gave me the idea of putting a factory back into the gut. So making a formula, a combined formula of strains that kind of work like a team in a factory to produce what you want them to produce. And so our first product we tried for my husband, John, is what we call Sugar Shift now, but the focus was to convert glucose and fructose in the body into mannitol, which we don't use, we eliminate, but it's a prebiotic fiber that feeds a broader population of good bacteria in the gut. And so, we made a small batch of it. We tried it with John, and he had some pretty significant improvement. We started giving it out to other people, with not just Parkinson's people, but just other friends and family. And they were like, this is the best thing we have ever tried. And so then we started to look for a way to understand how this team we had put together worked. And so we took some existing open source software and customized it ourselves so that we could put the genomes of the bacteria together in the software. So, for Sugar Shift, it has 8 different strains of bacteria. We have sequenced the genomes of every one of those strains. We put that into the software, and then the software has a database of about 17,000 different enzymes or metabolites. And that database came from the Argonne National Lab from all of their wet lab experiments of, what bacteria do with certain types of media or in combination together. And so then you can run that program for, 2 hours, 4 hours, 6 hours, 8, however long you want the consortium to work together to see how they work together as a team. What do they use and produce? So one organism's waste, so to speak, is the food for another one. And so we build these teams where they're working together in a sustainable way for about 12 hours. In the case of sugar shift, it's about 12 hours. And we were targeting the production of mannitol, but also increased production of butyrate, which helps the gut lining, and reduced glutathione, which is an antioxidant, and a number of other, we have increase in enzymes involved with detoxification. And I mean, I could go through a long, but so we took that concept and then kind of laid out about 25 different health problems and said, how can we build teams that will, for cardiovascular health, increase CoQ10 production and nitric oxide, or for sleep, how can we increase tryptophan and bacterial melatonin production? And, my chief scientific officer is a scientist by the name of Dr. Raul Cano, and he was a professor of microbial ecology at Cal Poly for about 35 years. I mean, he is world renowned for many different things. He was part of the team that sequenced the first strain of lactobacillus. He was a pioneer in using microbes to clean up oil spills, and a pioneer in what's called paleomicrobiology, where he revived ancient organisms that were trapped in amber occlusions and in deep sea cores from the ocean. And so, he has this incredible command of knowing what all these microbes' capabilities are. So, we sat down and mapped out, different women's issues or vitamin deficiencies or, anxiety. And, through his knowledge base and then our software, we take his knowledge base, put that into the software, run it and see how it looks. And then we can tweak it a bit. If it's not doing exactly what we want, then we sort of play with maybe a different strain or a different species and come up with these formulas that actually function to put back a function in the body.
Is really cool. So little bit of a two-parter here. How does your sugar shift affect diabetics? And then how do we know, or how do we get bacteria through stomach acid?
So the sugar shift probiotic, we actually did a clinical trial in diabetes, because even though I made it for John, there is evidence that Parkinson's may be what's called type 3 diabetes, or insulin resistance of the brain. But, Parkinson's is a tough disease to do a clinical trial for, because there's not clear endpoints. There's a lot of, the symptomology is not homogeneous. Yeah, homogeneous. And so it's just hard to measure because of the complexity of the disease. But since we knew it was affecting sugar, we said, well, let's do a clinical trial in diabetes. And we did that 2 years ago. Raul was born in Cuba and developed a relationship with a hospital back home in Cuba. And so we were able to do a relatively incredibly cost-effective clinical trial of 30 people on the sugar shift and 30 people on the control. And we did that for 90 days. And then at the end of the 90 days, we unblind. So it was a double blind. And at the end of the 90 days, we unblinded and took 10 people who had been on the product and kept them on for another 90 days. So we could measure some additional, because at 90 days, we didn't get a reduction in A1C, but at 180 days, we got a 14% reduction in A1C. And then So we had improvement in insulin, in HOMA-IR, which is an insulin sensitivity metric that's not used in the United States, but it's used worldwide. We got in the first 90 days, we got a stabilization compared to controls of fasting and postprandial blood glucose. But at the 180 days, fasting and postprandial blood glucose both got statistically significant decline as well. And then we measured something called serum endotoxin, or serum LPS. And that is a toxin from gram-negative bacteria. And when you have a leaky gut, that is crossing your gut and into your bloodstream, and it's very inflammatory. And that was actually one of the markers that we had the most significant decline in at both 90 days and 180 days was serum lipopolysaccharide, or endotoxin. And I think that's a really underappreciated contributor to a lot of our inflammatory diseases, because there are so many ways now that we're exposed to endotoxin, and people don't even know to look for it, and it's not a standard measure that's being done in your typical annual blood work. And so what was the other? You had a two-part question.
Just how we know for sure that it makes it through the stomach acid, or how are we able to get it past stomach?
So, well, in developing strains for the probiotic industry, they go through a number of screens to determine acid tolerance. Are they acid tolerant? Are they bile acid tolerant? And so most of your probiotic strains are going to have a certain level of acid tolerance that they've already been tested for. Now, you do get people so there are some products on the market who encapsulate because they want to get into the colon and past the upper GI tract. But we actually want our product working in the upper GI tract, because actually in the stomach and the small intestine is where fructose gets absorbed and that signal goes to the brain. And so we want that conversion starting right away. We don't, if you wait to the colon to try to convert fructose, it's too late. And I think you had asked something about colonizing. I mean, that's interesting too, because in our clinical trial, we did before and after microbiome testing. And, you don't always see the colonization of the strains themselves, but what you see is a change and a flourishing of the whole ecosystem. So we got an expansion in diversity. We got a restoration or an increase in keystone species like bifidobacteria. So we have 2 bifidobacteria in here, but there were other bifidobacteria beyond the ones that are in our formula. And so we got a increase in the relative abundance of bifidobacteria, an organism called Fecale prausnitzii, which is another keystone species, Roseburia eniolivorans, which is another keystone species. So, we got a number of these improvements in the overall health of the microbiome. And, what you're doing, I think, with a product like Sugar Shift is you're kind of shaping the terrain. So you're pulling out that excess glucose that feeds a lot of the pathogens, and that's allowing a broader, healthier microbiome to flourish.
Yeah, that makes sense. So have you seen anything in relation to H. pylori? Because that really kind of stood out to me when you said we didn't want it to pass the stomach. And that's fascinating, because I think that is a much, 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 much bigger problem than people realize, especially with the excessive use of proton pump inhibitors and acids. So have you seen anything with H. pylori?
Well, I mean, we haven't done specific tested focusing on H. pylori, but that's an interesting organism too. In Dr. Blazer's work, H. pylori he was an infectious disease doctor, and that was one of his areas of focus. And, initially antibiotics was the treatment for H. pylori once they determined that it was connected to ulcers. But, over time, his research has indicated, it's both good and bad. So it's another one of those things where it's got to function, and you don't want too much of it, but maybe you don't want zero of it either. Mm-hmm. So there's been and I can't remember exactly which side, there's an increase in stomach cancer under one circumstance and an increase in maybe neurological problems or something on it. And that's also interesting. I tell people all the time, like cancer and neurological disease are like opposite sides of the razor's edge. So people with Parkinson's, Alzheimer's, and Down syndrome do not get solid tumor cancers at the same rate as the rest of the population, but they have neurological disease. People with cancer have lower incidence of neurological disease. So they're kind of opposite sides of an out of balance immune system, I think.
Wow. Yeah, well, I've never heard of that or contemplated that, so that's fascinating. So, all right, let's start diving in. I want to start piecing this together. One of the biggest reasons that I wanted you is, a mutual friend, Petra. She's been trying to get me, I guess, to understand Barry's work or be exposed to Barry Nenem's work, and I feel like that is such a big key. So when I'd asked, Morley about Barry and his work, he said, you got to talk to Martha. So how are you tying all this together? Because for me, that's the biggest piece that I am having such a hard time wrapping my mind around, but I call it the lattice work of mental models. When you take great minds that are doing things and it overlays and you see these connections, whether it's Maury or Laszlo or Petra or Stephanie, I mean, there's so many amazing people that are putting pieces together, and then you with the microbiome aspect. So in the best way you can, this fundamental piece of Barry's work, how are you understanding it?
Well, it's interesting that you use the term the lattice work of mental models, because so Barry, maybe you've talked about him on your episodes before, but so Dr. Barry Nenem is a physical chemist. He founded the Department of Applied Mathematics at Australia National University. He is 88 years old. He has mentored 5 Nobel laureates, and he has done tremendous amount of work in colloidal chemistry, in structural chemistry, working with surfactants. There's so many layers of things. And he has a really interesting paper on COVID-19 and lung surfactant, which kind of turns the whole hypothesis of actually what was going on in COVID-19 in the lungs and in the physical body. And I got connected to him actually through a respiratory therapist from the United Kingdom who had been following some of the things that I write about sometimes on LinkedIn. And I have, on occasion, talked about this link with mycobacteria and the lungs and Parkinson's disease and mucus. And of course, mucus is a colloid, it's a colloid. And Barry's work is also related to the self-assembly of, self-assembling chemistry, basically. And alpha-synuclein is kind of a self-assembling polymer. And this, the prions and the amyloids and things are, they are assembling into these aggregates, which So that's sort of how all those pieces came together. But a lot of he has a book from a few years back, so he's definitely advanced the work since he did this book. But it's called The Language of Shape. And what I'll say, what he's been teaching me in his position that pretty much our understanding of the physiology of the body is wrong, kind of across the board. Like we've got it wrong because we haven't understood what's called the glycocalyx and the endothelial surface layer. And most people think of that, the glycocalyx and the endothelial surface layer, is a relatively new in the scientific field, but it's thought of more in the blood vessels. But really, this glycocalyx and glycobiology, glyco is the sugars, is all throughout the body, the extracellular matrix and these little, in the endothelial surface layer, and these tiny little cilia type hair structures that are allowing molecules in and out. And a lot of Barry's work is also around what are called nanobubbles, these tiny, tiny bubbles of O2 and N2 and CO2, and what those are doing in the body. And, what's so kind of crazy about that, I mean, I'm self-trained, so I don't have any of this dogma in my head. And, when he says, oh, well, gases have been ignored in all of science. And, then I start thinking about, like, once he shows you and you see something and then you start to read the papers, it's like, oh, wait a minute. They degassed that, or they're totally ignoring. I mean, not everywhere, but in a lot of places, they're ignoring these gases in the body. And I happen to know from my microbiome research in Parkinson's that there were also indicators of nitrosative stress or nitrogen stress, and that's one of the gases. And you think, okay, well, we are breathing in air. We talk about oxygen, we talk about oxidative stress. I mean, sometimes you hear people talk about reactive nitrogen species, but not that much. But, 78% of the air is nitrogen. So, it's like we've forgotten, or not forgotten, just totally ignored these really key pieces of the puzzle. And so, I've been fortunate to have been introduced to Barry, and then in that process, brought Petra and Laszlo into the picture, because in the self-assembly process, deuterium changes the stability of the structures. And so it not only changes, the speed of an enzyme reaction, but it also, if incorporated into a protein or a sugar or any of that, is going to make that structure harder to break. So that's sort of how
I want to
all
go back
these pieces started coming together anyway.
Yeah, and that's what it does, and then it makes you realize how much we don't know every time one of these layers comes on. But the way that I'm visualizing a healthy glycocalyx is like a wheat field, right? When you see like all the the iconic pictures of a wheat field blowing in the breeze, just all uniform, looking good, versus a hailstorm comes through and collapses it. That's going to be an unhealthy, which is allowing a lot of this to pass through unimpeded, or how it's supposed to be. So can you explain the significance in your terms? Because Robert Kiltz is the only one that I have ever heard talk about it. And I don't think that he was obviously like on Barry's level or been through. I think he's just aware of the glycocalyx. So how significant is this understanding?
Well, I'm going to do best job I can in my. So when you're talking about that wheat field, that is that endothelial surface layer. So that's those little hair-like structures. And then the glycocalyx is below that. So,
Okay.
and the glycocalyx is where there are these structures in the glycocalyx, and I equate them to something I call, well, I don't call, called research I've looked into called aquaporins, which are water channels. So, and they are, fluid channels. So basically, there are these sort of cubic structures that require surfactants. So the surfactants help build that structure. And there are different, so there are, surfactant protein A, surfactant protein B, surfactant protein C, surfactant protein D. And what happens with a lot of different, various things can be a sort of a wrecking ball to those structures. And so if it's like if you had I'm going to use Jenga. That's probably not a great example, but if you have a tower of Jenga blocks, say that's your glycocalyx, but there's, little, because in a Jenga block, you can have little squares in there. But if you start kind of throwing little balls at it or having something hit it, it starts to lose its structure and eventually it kind of falls over. And so this is actually where those nanobubbles of oxygen, nitrogen, carbon dioxide are flowing in and out through and into the blood vessels, or in and out of cells for that matter. And so when you don't have that proper structure, the things that need to get in and out cannot get in and out. And over time, the damage just gets worse and worse and worse. Does that make sense? I mean,
Yes,
I
but
have some papers here. I'm thinking, okay, do I have a picture here I could show you? I mean, I'm like, this is the stack of Barry's papers I'm sorting through.
this is where, and correct me if I'm wrong on any of this at any point, but this is where the importance of like the structured water comes into play, to where that we have the appropriate flow or structure of these epithelial in glycocalyx, right? Is that where? Because to me, and this is where I'm going back to the lattice work of mental models, this is where the light piece comes in. If to have proper function with the glycocalyx and the epithelial, hair-like structures, we have to have structured water, then that plays directly into the infrared light piece, right? And if that is the case, all of it is unbelievably connected. And now we can go back to the deuterium piece causing more problems because, the structured water, metabolic water, however we want to coin the term, or Pollock will call it easy water. I mean, I don't know how to articulate how important this comprehension is, but it's just like, it's almost like it's at the tip of my tongue that it's so close to right there. And Barry's probably would be screaming like, why do you not get this? But it's just like, I feel like it's so important, but it's just not completely coming together.
Well, so I'm going to go back on the water for a second because one of the very early things that Barry shared with me was a paper by a gentleman by the name of Mark Adams from 1948 that showed that you could inactivate bacterial viruses and proteins by shaking water and creating these nanobubbles. And by nano, but like people don't realize the size of the bubble. So we think of bubbles as like something that, visible size, but these are tiny, tiny little nano sized bubbles that behave in different ways. And so if you think about that, like you could inactivate the bacterial viruses and proteins just by shaking water. We do all this chemical stressor and stuff into our water supply. And, so anyway, that to me was kind of a big aha, this, shaking in the bubbles actually inactivating, viruses and proteins. And that's not how we treat our water, which is kind of crazy. So you can do these things with sort of bubble columns, if you will. So I don't know. I'm probably not the best person to like pull all these pieces together for you, but the bubbles coalesce, and like if you watch bubbles, like they'll come together and they'll meet and they'll enlarge and they'll pop. But certain of these nanobubbles have a stronger structure, and in Barry's work, a lot of this also has to do with the salt content.
Yeah.
So if you think, okay, we're sort of made of the ocean, our cells are of the ocean, and I think it's 0.17, I don't know if it's millimoles or what, but that ratio of salt content, if you go above or below that particular salt content, changes the coalescence of the bubbles and whether or not they hold structure or don't hold structure, and that is also quite critical in this whole process that's maybe not well understood. I think, people do understand that electrolytes are important, but I don't think they understand really this, delicate balance at a certain level of how that changes the mathematical structures that build the body. Yeah. Kind of back to the, glycocalyx and endothelial surface layer.
I'm just constantly blown away with how it almost always goes back to physics, and which is not my strong suit on any of this. And so it's just been challenging to get. You think that you have this base level figured out, and then nope, you're not even close, right? There's so many, and then the physics level is fascinating. And so Morley and I had been really just kind of going at him with the melanin piece, because the melanin is incredibly important. And once I was made aware that melanin will use light to separate water into hydrogen and oxygen, I'm like, wow, all right, this is part of Barry's piece, right? Because this is a mechanism at which we're getting these gases. And so again, I'm just trying to piece it together.
Yeah, so you, like in that you're getting gases, but like I always go back to the bacteria, which I know a lot better than the mechanisms. And although I have studied the melanin stuff in depth with Morley and with Herrera Solis's work, and really looking at what's going on with melanin in sort of a strange way. But
before we go back to where you were going on the melanin, what have you seen with Parkinson's? Is there any sort of connection with melanin and Parkinson's, especially say the substantia nigra? What is going on there?
So there's a connection to neuromelanin, which Morley and I have gone back and forth about. What's also interesting, my husband participated in a clinical trial very early on that was actually funded by the Department of Defense, and there were 800 subjects in the study, and it was supposed to stop the progression of Parkinson's. But one of the things that he had to do while he was in the study for 2 or 3 years, was go to a dermatologist every 3 months to make sure he didn't have a melanoma. And later I found out that the drug that they were testing was it was either a Janus kinase inhibitor or a tyrosine kinase inhibitor. Anyway, it had some mechanism in this melanin synthesis pathway, which is also interesting to me because melanin is also in the humans don't have a shikimate pathway, but bacteria do. And, melanin synthesis is in the shikimate pathway. So,
Fascinating.
but I always go back to bacteria because to me, as we're talking about nanobubbles back and forth, and we're talking about oxygen and nitrogen and CO2 and, what happens in the body with these things. But, the microbes are using nitrogen, using oxygen, using CO2. They're also producing them. So, because we have those computational models where, I've been looking at what they use and produce over the last 8 years, it's like, oh, wait a minute. This one makes dioxygen and these guys over here use the dioxygen. So I think that's another sort of missing piece of that puzzle is how the microbiome is dealing with gases in the body. And actually, my co-founder, Jack Gilbert, he's not involved with the company anymore, but he had a recent paper on, can't remember, one of the gases. And I was like, whoa, wait a minute. Jack's looking at the gas in the microbiome. And he has looked at that before and what goes on, say, when you do a GI surgery. And so you typically don't have oxygen in the GI tract, but once you cut it open to do the surgery, you're letting in oxygen and nitrogen and, air. And how does that rapidly change the microbiome and what happens there? So, I continue to always sort of go back to what the microbes are doing in relationship to the terrain that we've put in place, whether that's, with the water, with the food, with the chemistry, and how that's changing our physiology and chemistry.
Yeah. The shikimate pathway in the bacteria, that's exactly how glyphosate kills plants. So, and I guess acts as an antibiotic. But all right, we're gonna have to circle back to the melanin at a later time because you got my mind just racing, and it would have had to have been a tyrosine inhibitor that he was given, and that it's my understanding that melanoma is much more directly related with basically a lack of infrared and a lack and hyper UV. So like all these blue lights and stuff that we've got, no sunshine. So I find that fascinating.
So we also, so back to Raul for a moment in my microbes or his microbes. He has a microbe called Micrococcus luteus, and it makes a yellow pigment, but it's been used, I mean, not his in particular, but that species has been used in sunscreens because it has a lot of genes involved in UV protection, but it also has a lot of DNA repair genes. And I think, not only have we lost the microbes in our gut, but we've lost these microbes on our skin that were also providing us with a lot of information and protection. And, another interesting thing about that microbe is it can actually clean up radioactive nucleotides. So it'll take radioactive material and incorporate it into its cell wall and then clump up in a cluster so that you could remove it from water. So, Raul did a proposal to the government of Japan after Fukushima to help with the cleanup, but it didn't get selected. Anyway.
Okay. Well, on this gut microbe, I guess we got the microbiomes everywhere, but where is the significance of the fungal component of all this? Because we talk about bacteria, right? And then I feel like that's, like with Barry and gas, that it gets emitted a lot of times, or Jack Cruz, how he'll emphasize light. We don't talk much about fungus unless it is in a negative context,
right?
Or I guess Saccharomyces boulardii is actually fungal. But what is your take on that?
Well, so I have a book called The Life Bridge. The way to longevity with probiotic nutrients. And there's a lot of discussion in there, but one of the about how microbes make inorganic matter living matter and make minerals more bioavailable to us. But there's a section in there on selenium in particular, and they talk about brewer's yeast or nutritional yeast. So when you ferment in a yeast, the selenium that was like a USP, just inorganic selenium, we can't absorb it very well. We've absorbed a very small amount of it. But if we're getting selenium from a fermented yeast, a brewer's yeast, that selenium is like 268 times more bioavailable than the inorganic compound. And so it's complexing these minerals in ways that make them enzymatically active and able to be absorbed in the body. But one of the comments that they make in the book that I mentioned to Morley that kind of blew his mind a little bit was yeast is a mini mammal. So yeast is, we're a mammal. And yeast is actually one of the models used in scientific research often to study different mechanisms in the human body because it is a mini mammal. And it's used a lot in the study of copper, which of course is, Morley's, one of Morley's big things. So we've learned a lot from looking at yeast studies with copper. So, I think again, part of our problem in our food supply is lack of diversity. So, backtrack 100 years ago when people made bread, they were making their bread with basically wild yeast. So, somebody was sharing sourdough with them, or they were leaving mixing the bread with water and salt and sugar and leaving it out to pick up the wild yeast from the air and leavening the bread that way. And so you were getting a much wider variety of fungi and bacteria from wild fermentation of the local environment. And what we have now is, everything's been commercialized and standardized and narrowed down. So, everybody's using the same brewer's yeast or the same, baker's yeast or the same, 12 strains of probiotics. For the most part, the vast majority of probiotics that you will find in a store are coming from just 2 or 3 vendors. And so if you look on the back of them and it says Lactobacillus plantarum or Bifido longum, there's a handful of people who produce that, and it's not, you're not getting a big variety. So you're not getting a huge variety if you get, this bifidolongum and that bifidolongum, they're very likely might be the same strain.
Okay. Well, all right. Where do we send people to learn more about your company and Sugar Shift? That sounds fantastic. I'm going to dive into that a lot more. I know Morley's mentioned it a few times, but like I haven't fully just jumped down that. So I think that's incredible. So where do we go to learn more?
So you can go to our website is biotiquest.com. That's BIOTIQUEST.com. And that has all of our formulas on there. If you want to know about my Parkinson's work, you can go to Martha's Quest. And I write a blog about alternative health things related to Parkinson's in the microbiome. And I post all of my podcast interviews on there as well. And if you want to know more about the collection of poop and the history behind what we did to get to this point, you can go to thebiocollective.com. And then we actually have a fourth company that we didn't talk about at all, but that works on the agricultural side, making soil-based communities of microorganisms to clean up glyphosate, increase yield, and restore the vitality of the soil. And that is ancientorganicsbio.com.
Well, Martha, I had no idea. I completely dropped the ball because that is really the regenerative agriculture. It's like cancer. So I own a farmer's market, a grocery store, and it's all about the local food regeneratively. And that tool is massive. So we are going to have to visit more about that because that's a big deal, especially here in Arkansas. They use a lot of glyphosate.
Well, I would love to come back and talk to you about all the work we've done there over the last 5 years, studying our product in everything from, just like vegetables to alfalfa to cotton to corn and soy to wine grapes, and, showing what we can do to improve the soil quality, improve the yield, but get the benefit
Amazing.
of cleaning up the organophosphates.
Amazing. Well, Martha, thank you so much for taking the time to visit with me.
Well, thank you for having me. I appreciate it, Logan.
Thank you for joining us on Sowing Prosperity. Be sure to follow along across the social media platforms, including YouTube, and be sure to go to sowingprosperity.com.